Good morning. My name is Manon. I am a member of the board of the International Society of Vaccines and incoming president. I'm also chairing our committee, which is responsible for industry relations. And one of the ideas that came up during our discussions was that it would be really nice to discuss topics that were of specific interest to the industry. And I'm really excited to share today the first Industry Interaction Committee seminar. We have decided to have this first virtual symposium on pneumococcal vaccine, and we've been able to put together a wonderful panel of speakers. And we hope that with these speakers, we're going to give you a global overview of what is going on in the field of pneumococcal vaccines. So with that, I would like to invite our first speaker, who is Kathleen McElwee from Pfizer. She's responsible, she's the clinical global lead for the PCV20 program at Pfizer, and she will discuss with us the impact on pneumococcal vaccines, of vaccines on pneumococcal disease. So with that, Kathy, I'd like to invite you to share your screen, and we're looking forward to your presentation. And by the way, for the audience, it would be great if you could type your questions in the Q&A, and then I will make sure that the questions are going to be relayed to Kathy. Thank you, Kathy. The floor is yours.
Thank you, Manon. Good morning, everyone. My name is Kathleen McElwee, and I'm a Director of Vaccine Clinical Research and Development at Pfizer. I am also an infectious disease physician. It's a pleasure to speak with you today about a topic that remains central to global health efforts, the impact of vaccines on pneumococcal disease. Here are my disclosures. My goal is to provide a broad overview of pneumococcal disease by discussing the bacterium Streptococcus pneumoniae, examining the current disease burden, tracing the historical evolution of PCV7 through PCV20, discussing the profound impact vaccination has made so far on children and adults, and discuss future directions. Streptococcus pneumoniae is the bacterium responsible for pneumococcal disease. Early in the 20th century, researchers identified multiple serotypes that were each characterized by distinct capsule polysaccharides. The outer polysaccharide layer is what defines the serotype, functions as a virulence factor, and acts as a vaccine target. Over 100 serotypes have been identified, though not all are equally pathogenic. Predominant serotypes can vary by age and geographic region. It is only a smaller portion of these 100 serotypes that tend to cause the majority of severe disease. Streptococcus pneumoniae is commonly carried in the nasopharynx and is transmitted by respiratory droplets to other individuals. Pneumococcal disease represents a family of infections caused by Streptococcus pneumoniae. The panel on the left reflects important presentations of pneumococcal disease as defined by the anatomical area. These infections can be classified as invasive and non-invasive disease. Invasive pneumococcal disease, or IPD, is defined as the presence of the bacteria in a normally sterile site, such as cerebrospinal fluid, pleural fluid, and blood. It is the leading cause of the most serious cases of pneumococcal disease, which includes meningitis, sepsis, and bacteremic pneumonia. Non-invasive disease includes non-bacteremic pneumonia and otitis media. As noted in the panel on the right, bacteremic and non-bacteremic pneumonia is a major contributor to pneumococcal disease. Worldwide, over 700,000 all-cause pneumonia deaths occur annually in children less than 5 years of age. Approximately 100,000 U.S. adults are admitted for pneumococcal pneumonia hospitalizations yearly. And moreover, the prevalence of non-bacteremic pneumococcal pneumonia is more than 15-fold greater than that of invasive pneumococcal disease in U.S. adults over the age of 50. These statistics would be even more dire without vaccines. as evidenced by epidemiologic surveillance in the United States, known as the ABCs, the Active Bacterial Core Surveillance System, run by the Center for Disease Control and Prevention in the US, the youngest and oldest age groups continue to have the highest incidence of IPD. In children less than 1, the incidence was 10.8 per 100,000, decreasing in older children and adolescents, then increasing in adults with a peak incidence of 27.4 per 100,000 in adults 85 years of age and older. Vaccines have been targeted for both pediatric and adult age groups. As children are one of the highest risk groups, conjugate vaccine technology was a breakthrough for children less than 2 years of age, where polysaccharide vaccines are not effective. Linking a polysaccharide antigen to a protein carrier improves the immune response from a T-cell independent process into a much more potent T-cell dependent one by activating B, T, and memory B cells. Whereas both activate B cells to produce antibodies, a key difference is that conjugate vaccines induce T-cell dependent immune responses that can be boosted. Conjugate vaccines also induce memory B cells and mucosal activity. It now marks 25 years since the first conjugate vaccine, PCV7, was licensed in children in the US in 2000. It contains 7 serotypes, which were among the most prevalent and invasive before widespread adoption and revolutionizing childhood immunization. Dramatic declines in IPD were seen among vaccinated children, and though the vaccine was not licensed for adults, reductions in IPD and pneumococcal pneumonia were observed through herd protection. 10 years later, in 2010, PCV13 was first licensed in children to address non-vaccine serotype burden of disease, and later had a subsequent age expansion to adults 65 years of age and older, and then to adults with certain immunocompromising conditions. PCV20 was licensed in 2021 for adults, followed by licensure for children. PCV20 added 7 additional serotypes as conjugates. As we see, this is also a 25-year timeline with an overview of the introduction of other pneumococcal conjugate vaccines, with the year indicative of the first licensure for either the pediatric or adult indication. To demonstrate the impact of PCVs on IPD in children less than 5 years of age from 1998 to 2015, before and after implementation of PCV7 and PCV13, this is a graph of cases of IPD in the US as observed in the active bacterial core surveillance. The impact of PCV7 was remarkable. Within a few years of its introduction in the United States in 2000, followed by PCV13 in 2010, a rapid and sustained decline of 91% in overall IPD incidence in children aged less than 5 years was observed from 1998 to 2015, from 100 cases per 100,000 in 1998 to 9 per 100,000 in 2015. During this same time period, IPD due to PCV13 serotypes decreased 98%, from 91 cases to 2 cases per 100,000. The introduction of PCV7 and PCV13 into the routine pediatric vaccination schedule in the United States has also had an indirect impact on IPD in adults. Children are the primary reservoir of Streptococcus pneumoniae. Therefore, the reduction in nasopharyngeal carriage of vaccine-type strains in vaccinated individuals results in subsequent decreased transmission to unvaccinated populations. This decreased transmission has led to the observed reduction in rates of IPD in adults. When used as part of a national immunization program, population protection is a result of both direct and indirect protection. PCV13 has demonstrated a significant impact on all-cause invasive pneumococcal disease in multiple countries around the world that use different vaccination schedules in children. For each of these countries, the overall reduction is shown with the age range below. Overall, there was a reduction of 60 to 82%, with some of the highest reductions in the Gambia at 80% in children less than 5 years of age, and Australia at 82% in children less than 2 years of age. When we consider the global impact, the figures are impressive. In a study using data from the Global Burden of Disease Study and Modeling, it estimates prior to the introduction of PCVs in 2000, there were approximately 600,000 deaths due to pneumococcal disease in HIV uninfected children less than 5 years of age. By 2015, pneumococcal deaths in children less than 5 years of age declined by 51% to 294,000. Despite the considerable reductions observed from 2000 to 2015, total pneumococcal deaths remained a substantial cause of mortality in HIV uninfected children less than 5 years of age, accounting for at least 11% of all deaths in that age group in 2015. The majority of these deaths occurred in low and middle income countries with the highest disease burden. I have shown the graph on the left earlier in this talk to mention that in the United States, the youngest and oldest age groups have the highest incidence of invasive pneumococcal disease. The Advisory Committee on Immunization Practices, the ACIP in the United States, has recently changed the age recommendation in older adults in October 2024 by reducing it from 65 years of age down to 50 years of age. Recommendations for adults 19 to 49 years of age with a risk condition have remained unchanged. This updated recommendation is expected to improve vaccination rates among adults 50 to 64 years of age, as an estimated 32 to 54% of many adults in this age group have at least one risk condition but remain unvaccinated. Lower respiratory infections, which include pneumonia, contribute to a substantial burden of disease worldwide. According to the Global Burden of Diseases, Injuries, and Risk Factors Study in 2017, there are more than 10 million cases of lower respiratory tract infection across all ages in 2017. In 2021, the WHO released the top 10 causes of death globally, and LRTIs were the fifth leading cause of death worldwide and remain the world's most deadly communicable disease other than COVID-19. Common causes of LRTIs, like pneumonia, include Streptococcus pneumonia, but also influenza and SARS-CoV-2. An especially important PCV13 efficacy study in older adults, known as the CAPITA trial, Community-Acquired Pneumonia Immunization Trial in Adults, demonstrated that using the conjugate vaccine in individuals 65 years of age and older significantly reduced vaccine-type pneumococcal pneumonia. It is one of the largest double-blind, randomized, placebo-controlled vaccine efficacy trials ever conducted in older adults. Over 84,000 adults were enrolled throughout the Netherlands and followed for a median duration of 3.93 years. Key exclusion criteria and demographics of individuals with comorbid conditions are shown. 42.3% of patients had one or more comorbid conditions that increased their risk for pneumococcal disease. The results of this study are shown here. For the primary endpoint in the dark blue arrow to the left, PCV13 demonstrated vaccine efficacy of approximately 46% against a first episode of any vaccine-type pneumococcal community-acquired pneumonia. In the middle light blue arrow, you can see the vaccine efficacy against first episode of vaccine-type bacteremic non-invasive CAP was similar to that seen in the primary endpoint at 45%. In the gray arrow to the right, PCV13 showed a greater protection against first episode of vaccine-type IPD with vaccine efficacy of 75%. The CAPITA study underscored not just protection against invasive disease, but also a measurable reduction in non-bacteremic pneumococcal pneumonia among older adult populations. Despite these successes, after the introduction of PCVs, the highest burden of pneumococcal disease has historically been due to serotypes not included in licensed vaccines. The phenomenon of serotype replacement has become a growing concern. By reducing disease from those included in conjugate vaccines, other serotypes not covered by the vaccines have had an opportunity to emerge as new leading causes of disease. This slide demonstrates the percent of IPD cases with serotype coverage for PCV13 and PCV20 in the US from 2019 to 2020 in children less than 5 years of age on the left, and on the right in Europe, 2019 for both children less than 1 year of age and children 1 to 4 years of age. The 7 additional serotypes included in PCV20 are anticipated to address an additional 40% of IPD in the US and approximately 1/3 of IPD in European children less than 5 years of age. The 7 additional serotypes were selected based on their relative prevalence as a cause of IPD, their generalized geographic distribution, and other factors that would support inclusion, such as the presence of antibiotic resistance, association with outbreaks, and greater disease severity. PCV20 marked a significant step in the strategy to combat serotype replacement by broadening serotype coverage, building on the now 25-year legacy of PCV7, followed by PCV13. When PCV13 was developed to expand protection against 6 additional serotypes from PCV7, it was not considered feasible to perform an efficacy trial. Immunogenicity bridging comparing PCV13 to PCV7 was used to support licensure globally. This same approach was used for PCV20, which is now the broadest conjugate vaccine licensed in both adults and children. In the adult vaccine trials, the 7 additional serotypes were also bridged to the 23-valent pneumococcal polysaccharide vaccine. Beneath both PCV7 and PCV13 is the data available for each. And while we have discussed real-world effectiveness and impact for PCV7 and PCV13, work to assess this impact for PCV20 has begun now that PCV20 has been implemented. This is a map by the World Health Organization of all pneumococcal conjugate vaccines, demonstrating the introduction of PCVs across the world. This data is collected from national authorities regarding national immunization programs. It may not reflect the additional reach of vaccines on the private market. While there has been widespread uptake, certain regions have not incorporated PCVs into their national immunization program. Despite the substantial burden due to pneumococcal disease that persists across age groups, global pediatric immunization against pneumococcal disease remains well below rates observed for other common vaccine-preventable diseases, such as diphtheria, tetanus pertussis, polio, hepatitis B, and Haemophilus influenza type B. According to the World Health Organization, global coverage with 3 doses of pneumococcal immunization in children was estimated at 65% in 2023. While this percentage has increased over prior years, there remains room for improvement. This is a map of the current areas where PCV20 is recommended in adults, as seen on the left, and children on the right, as we aim to bring PCV20 to the rest of the world. We have completed a PCV20 clinical trial for adults in India, where we also have an ongoing study for pediatrics. As we reflect on the past 25 years and the history of PCV7 through PCV20, while pneumococcal disease remains a global health concern, there is also much to celebrate. Additionally, we look towards the future, and for pneumococcal conjugate vaccines, the timeline continues on as Pfizer is currently investigating a 25-valent pneumococcal conjugate vaccine candidate. In summary, pneumococcal conjugate vaccines have been impactful at reducing disease in both children and adults. Higher valency vaccines have been developed or are in development to address non-vaccine type disease. Vaccination remains one of the most impactful public health tools and remains our best bet in reducing the devastating impact of this bacterium. Thank you for your time and attention. I look forward to any questions you may have.
Thank you very much, Kathy, for a beautiful overview. And I would like to congratulate Pfizer for developing this vaccine. It clearly has had an amazing impact. I want to invite the audience to type their questions in the Q&A box. And meanwhile, I would like to start with my first question, because you speak very clearly about, serotype replacement, and that's obviously a big concern. My question relates to how multivalent can you make this vaccine? And is that indeed the best path forward, or do you see other opportunities to create a vaccine that might be broader protective?
How broad can a vaccine go? I think that's the question we're all trying to answer. And I think in vaccine development, it is a great question. Whether or not the sky's the limit, or if there's limitations as far as how big a vaccine we can make. I don't think I have the best answer to that. But I think, just trying to use epidemiologic surveillance to survey what serotypes are the most troublesome and the highest impact in each region really helps you develop what would be the most important in those places.
Yeah, your response actually leads very nicely into one of the first questions we see in the chat, and that is from Xavier Lawrence, who basically asks, how does Pfizer plan to increase serotype 3 effectiveness in future vaccines? Because as you put in more serotypes, my understanding is that sometimes the immunogenicity to certain already present serotypes might actually go down. So
Yes.
how does Pfizer plan to increase the effectiveness against serotype 3?
Yes, I don't. Serotype 3 has been a problem serotype, and I have nothing to say about how we plan to do it. The 25-valent vaccine will include serotype 3.
The next question relates to the safety data on the 20-valent vaccines in children and adults. How much data is there available, I guess the question is.
Data for safety? Well, PCV20 in clinical trials included over 3,000 children in just the core trials. And PCV20 was seen to be just as safe as PCV13, had similar safety profiles. And now that the vaccine has been implemented, in many countries, I believe, it's been probably in the millions of doses that have been given.
So a question from Kishore Alugupali is how many immunizations are needed, primary and boosters? I guess that might be different in different countries, right? The recommendations.
The recommendations are different in many different countries, and so as shown, we've seen that it has been effective at different immunization schedules, and so the recommendations are by country and by organization, national immunization programs.
Right. The next question from Jeroen Langerijs relates to the polysaccharides conjugate vaccines have been extremely successful, as we've seen in your talk, but serotype replacement remains a problem. Would a protein-based vaccine be a possible alternative?
I think, we don't know.
It relates to my first question too. Like, what else can you do?
Yes.
Yeah.
I believe that Cynthia Whitney will probably address some of these challenges in her talk, but I don't know her exact content. Yeah.
Right, so we have a ton of more questions. So I'm going to just pick a few, and then the rest, I would like to say, people have asked, are you willing to share your slides? And then perhaps we can, people can reach out to you with their specific questions that we really cannot have the chance to address today. So is that a possibility, Kathy? Would you be willing to share your slides?
Sure.
Okay, so are you planning to explore the addition of adjuvants?
I have not, we haven't discussed any adjuvants in the clinical development program for PCV20. There's
Okay. Do you expect the future use of PCV20 in a 3-dose schedule rather than the 4-dose schedules, particularly outside the US, such as in Europe? Question from Rohan Wilms from Inventprice.
I'm sorry, can you repeat the question?
Yeah, he asks whether you expect a future use of PCV20 in 3-dose schedules rather than the 4-dose schedule, particularly outside the US, in countries, for example, in Europe.
Yeah, the schedules of the vaccines is really up to the regulatory authorities and those recommendations by, you know, advisory committees and national immunization programs. So, you know, what they do is based on their analysis of the data.
Right. So, Kathy, I would really like to thank you for your contribution for this wonderful talk. There are lots of questions in the Q&A. Maybe you can take a look while we're going to ask Dr. Desmet to continue with her talk. And again, thank you to the audience for being so engaging and asking so many questions. And I'm sorry that I wasn't able to run through all of them. I'd like to start with the introduction of Stefanie Desmet. She is the head of the National Reference Centrum of Pneumococci in Leuven, Belgium. She's also a professor and assistant professor at the University of Leuven. And what she will do today is she will discuss the trends in pneumococcal epidemiology in children following implementation of pediatric immunization program. Thank you, Stefanie. The floor is yours.
Thank you. I have just one thing to do.
As you are preparing to share your slides, are you also willing to share your slides following this presentation?
Yes, of course I am. Yeah.
Wonderful. Thank you.
Okay, I hope I'm now ready. You can see my slides.
I can't see your slides yet.
You can't see them? Okay.
No, I still see you. Yeah, wonderful. They're coming.
I hope
They're
you
there.
can
Thank
see
you.
them now. Yeah.
Yes.
Okay. Okay, hello everybody. First of all, thank you for the introduction and also for the invitation to speak here on this online symposium. As already explained, I will try to give you an overview of some real-world trends observed in pneumococcal epidemiology, both in children and adults. And I will mainly focus on countries with mature pediatric immunization program. Please find here my disclosures. So in this presentation, the focus is on changes in pneumococcal epidemiology after the introduction of PCVs in childhood immunization programs. So it was already shown by Kathleen that the implementation of PCV10, PCV13 results in changes in epidemiology. And this is due to, on one side, the direct effects in the target group, here in the children, but on the other side, also the indirect effect in other age groups or risk groups, as we know that there is not only prevention of infection, but that these vaccines reduce also the nasopharyngeal carriage of pneumococci, which is of course important for the transmission of pneumococci to other risk groups. So we know that if less vaccine serotypes are carried, less transmission will occur, and we will get an indirect effect of the vaccination. And I will mainly focus here dig into more detail into the serotypes and look into the changes in vaccine serotypes and non-vaccine serotypes, and mainly PCV13 and non-PCV13 serotypes. I will not talk about impact of adult vaccination on pneumococcal epidemiology, which is of course also very interesting, but this is out of the scope of this presentation. So next to changes in epidemiology due to or following vaccination of children, we were also recently confronted with another intervention that resulted in a significant change in pneumococcal epidemiology, and it was the COVID-19 pandemic and its associated containment measures. So this resulted, the COVID pandemic and containment measures, in a drastic reduction of the invasive pneumococcal disease cases. And the effects of this drastic reduction on the long term need some attention, and I will discuss some recent data regarding the rebound in pneumococcal infections and also the impact of COVID-19 on the serotype distributions. And during my talk, I will use my own country, Belgium, as an example of, let's say, a high-income country with a mature PCV program. So you see Belgium, a small country in Europe, and we introduced in 2007 the 7-valent vaccine in our childhood immunization program in a 2+1 dosing scheme. And we rapidly had a high vaccine uptake that remained stable until today, more than 92% of the children. And like a lot of other countries, we switched after 4 years of use of PCV7 to a higher valency vaccine, which was PCV30. But during a period of 4 years, we interrupted the use of PCV13. We used PCV10 instead of PCV13. And this way, Belgium is seen as a sort of an example to study the impact of childhood vaccination, and more specifically, an example to study the impact of a change in a childhood vaccination program and the impact of that change on pneumococcal epidemiology. Now, first we look into IPD epidemiology in Belgium. So here it's in Belgium and in the children, and we focus on the youngest children with a high pneumococcal disease burden. And in this graph, the total IPD cases are indicated by the dark blue line over a period from 2007 to 2019. And the different colors in the bars indicate the serotypes responsible for this IPD, grouped based on their inclusion in vaccines. And after the introduction of PCV7, and I need to say that in Belgium PCV7 was already used also some years before, it was not officially in the childhood immunization program, but after introduction of PCV7, it's clear that the PCV7 serotype IPD decreased over time. But total IPD, here we can see that it was only after PCV13 introduction that you can see a steep decrease in total IPD due to a decrease in PCV13 serotype IPD. In 2015, let's say the lowest number of cases caused by vaccine serotype IPD. Now, important to say is that we could not eradicate by all vaccine serotype IPD. So like in other countries, we still had some residual disease caused by vaccine serotypes, but we had a very broad effect on invasive pneumococcal disease with a decrease in IPD cases and mainly non-vaccine serotypes that caused at that moment the disease. Now, in 2015 and also 2016, we switched to that 10-valent vaccine. And what happened was, let's say interesting to see is that we observe again an increase in vaccine serotype IPD. With the orange bar, these are PCV13, non-PCV10 serotype IPD that again increased. And this was due to the increase of one specific serotype, serotype 19A. 19A included in PCV13 and not in PCV13. This illustrates that a stable, strong immunization program in children is needed, and that we need to maintain the pressure or the suppression of important vaccine serotypes. And that when you do a change in a program, like here is change from a higher valent vaccine to a lower valent vaccine, that this may impact on the incidence of IPD, and that some vaccine serotypes that are, let's say, only rarely observed, that they can re-emerge and become again important. And of course, that the increase of that serotype 19A IPD and resulting in an increase in total IPD led us switch back to the 13-valent vaccine in Belgium. So this is a Belgian situation, but I want to give you also a more global view on the epidemiology of IPD after introduction of PCV10 and PCV13. And I'm thankful to be able to share some data of the Serenade project. These data were recently also published in the Lancet Infectious Disease. And interesting is that in this project, data from surveillances from over the world, from sites with extensive use of PCV13 or PCV10, were analyzed to define the impact on both incidence and serotype distributions of strong childhood immunization programs. And for the evaluation of the change in incidence after the introduction of PCV13, data from 31 sites were taken into account. Incidence rate ratios were calculated. You see them here, incidence rate ratios. And an incidence rate ratio equal to 1 indicates no change in incidence compared to the pre-PCV period. And an incidence ratio below 1 indicates a decrease. An incidence ratio above 1 indicates an increase in incidence compared to pre-PCV. Now, after 6 years, a decline in vaccine serotype IPD was observed by 94 to 95%. You can see these are the same numbers as Kathleen already has shown. With the same incidence rate ratio for countries that previously used PCV7 before they introduced PCV13 and PCV10. But an interesting conclusion was that serotype 3, which is included in PCV13, did not decline. And this will come further also in the presentation, that serotype 3 is a very important serotype, vaccine serotype. Well, we see that in the real world epidemiology that we get no decrease of that serotype 3 IPD. Now, we have a resulting proportion of about 26% of IPD that is still due to PCV13 serotypes when we look into data from different countries. Now, due to serotype replacement, an increase in non-PCV13 serotype IPD was observed, with some heterogeneity between the different countries. And taking these two together, so a decline in vaccine serotype IPD and a more smaller increase in non-PCV13 serotype IPD, results in total decrease of invasive pneumococcal disease with after 6-year decline, and when we here focus on PCV13 sites, of decline of 67 to 79%, depending on the fact of that there was before a substantial use of PCV7 or not. Now, this is regarding the impact on incidence. When we focus more on the serotypes and the serotype distribution, also in that Serenade project, data from different countries were taken together. We need to stress that these are mainly high-income countries with half of the size from Europe or North America. And what we observe is that the most important serotypes are serotype 3 and 15BC. So serotype 3, as you can see, it's a PCV13 serotype, is the most important one. And we also have some residual disease of serotype 19A, which is also included in PCV13 and 19F. So the serotypes with the red shade are serotypes that are included in PCV10 or PCV13. There are some differences between countries, and this can be also given an example. So for example, in Belgium and England, I indicate some data. So 5 years after the introduction of PCV13 in both countries, then we can see that the top 5 predominant serotypes in children less than 5 years are different. So in England and Wales, in the top 5 with 15 BC and serotype 8. These two serotypes are not in Belgium in the top 5, but 23B and 33F are in the top 5 at that moment. So again, this stresses that there is some local epidemiology that needs to be taken into account, and that serotype distributions may vary between countries. Now, all these data from the Serenade study come from surveillances and from the period pre-COVID. But of course, what is now the impact of COVID on the incidence and serotype distribution? Now, when we look into the Belgian situation, like it's the same observed worldwide and described also by the IRIS study group from Angela Brugman, a decrease in pneumococcal IPD was observed and a rebound since half of 2022, 2023 was observed, and we get again same levels as observed pre-COVID, and in Belgium we even have higher levels than during the period where we used the first time PCV13. Data from other countries, this is England. Here you can see the impact of COVID also on the IPD in children. And I need to say in England, they did also at the same time in 2020, the switch from 2+1 to 1+1 schedule. But it's clear that also COVID-19 here had an impact with a decrease in IPD cases with then a rebound back to pre-COVID-19 incidence rates. Now, when we focus on the serotypes that are responsible for both vaccine serotype IPD in Belgium and non-vaccine serotype IPD, and the recent evolution after introduction of PCV13, so the different colors indicate different periods. So the first is the first PCV13 period. In gray, this is 2 years in the PCV10 period, and then 2022 and 2023 is post-COVID and after reintroduction of PCV13. Then it's clear when we look into the PCV13 serotypes, that 19A is still the most important serotype, and that during the PCV13 period, we had very high numbers of that 19A, which was also the cause of the increase of total IPD, with again a decrease post-COVID and after the switch to PCV13. Regarding the other vaccine serotypes, there is serotype 3 IPD in this youngest age group, and there was a slight increase in serotype 14. Now, regarding the non-PCV13 serotypes, you can observe that there are a lot of different ones that cause invasive disease. And also there we can observe some evolutions. Striking is that 12F, which was the most important non-vaccine serotype in the period before COVID, that then we observed not only in Belgium, but in a lot of other countries that serotype 12F decreased. On the other side, we have evolutions with an increase of 24F, increase of 23B over time, and then some serotypes that increased, although these are lower numbers after the COVID-19 pandemic. But we didn't know what to expect after COVID, and we can say there are some changes, but mainly we see that we have a rebound back to serotype distributions that were observed before the COVID pandemic. And this is true for Belgium, but also for other countries worldwide. Oh, this was the part regarding children. Now, when we look into the incidence in adults, this is also a graph now with Belgian data with incidence of IPD over a period of time. Then you may observe that after the introduction of PCV13 in the childhood immunization program, we saw a decrease 2 years later of the incidence, with then some stabilization and even a trend of, again, an increase of the IPD incidence in adults. Belgium, again, interesting to look into detail because we have a low and stable vaccine vaccine uptake in the adult risk groups, which is of course important to look into potential impact of childhood vaccination. England, here, when we look into all ages and evolutions in all ages, after PCV7, a decrease in total IPD and a further decrease in after PCV13, but then let's say a lowest point, attaining a lowest point, and again a trend of an increase of IPD. And also in the Pseronate study, this was studied in detail. Here you can see the data of the adults aged 65 years and older, and again the same graphs, the evolution of PCV13 serotype IPD and non-PCV13 serotype IPD, with in adults, in these older adults, a decline by 74 to 77%, but also here serotype 3 did not decline, and PCV13 serotypes were still responsible for an estimated 29% of IPD. Also for the non-PCV13 serotypes, so here we also observe that increase over time, with again some heterogeneity between countries, leading to an impact on all serotype IPD, a decrease by 22 to 27% in the adult age groups, but also there some variation between countries, and also in some countries, let's say after 5 years, let's say attaining a plateau and a trend to again some increase of IPD. Now, when we then focus on the serotypes that cause that IPD in adults, again, data from the Pseronate study, serotype 3 is topped as the first one, and to a lesser extent also vaccine serotype 19A and 19F persists. But compared to the serotype distribution in children, serotype 8 and 9N ranked in the top 5, and serotype 15B and 23B, which are very important in children, are not observed in the top 10 for adult IPD. Then here's some data from Belgium with the evolution. Again, the 3 periods, PCV13, PCV10, and then post-COVID and PCV13 again. And there we observed some interesting evolution regarding vaccine serotypes. So serotype 4, which is was already included in PCV7, increased post-COVID. Again, something that was not only observed in Belgium, but also in our neighboring countries like the Netherlands and England. For the other serotypes, the PCV13 serotypes increase in serotype 3, 19A highest proportion in the PCV10 period, and then some other evolutions like 22F, 8 that increased over time, and then 12F that significantly decreased during the COVID pandemic. So it's clear that we discussed change in epidemiology in the youngest children and adults, and it was based on these data and the data from the Pseronate study that the serotypes that cause disease in children are not the same as the ones that cause disease in adults, but also within the adult age groups, we see some differences regarding the serotype distribution, with some serotypes that are seen more in the younger adults, observed more in the younger adults, and some serotypes that are more observed in the older adults, which makes that when countries need to decide about the implementation of a certain pneumococcal vaccine, that they need to take into account the local epidemiology and of course also the serotype distribution and the target group for vaccination. And I give here an example of Belgium where there is an advice for the adults aged 65 years and older. Then we need to look into what is the theoretical coverage of the IPD cases by the different vaccines, and this helps decision makers to decide, and this is only part of the decision, of course, what to implement in their childhood, in the different immunization programs. So I'm a bit over time, but with this slides, I want to conclude, and I hope it's clear that you all saw that in countries with a mature childhood vaccination program, that vaccine serotype IPD decreased both in children and in adults, so in adults due to the indirect effects, and resulted in a decrease in total IPD incidence. There is some there are problems regarding serotype 3. So serotype 3 pneumococcal disease did not decrease, although it's included in the vaccine. This serotype is the most important serotype causing IPD both in children and adults. So further investments are really needed to develop strategies to prevent serotype 3 IPD. And other vaccine formulations than the ones we have now available are probably needed. There are age-related differences and country-related differences in serotype distribution. So there is a need for strong surveillance to help decision makers in the context of availability of different pneumococcal vaccines. And I show with the Belgian example that a decrease of vaccine pressure in children, so when you change the vaccine or we change the number of doses given, that this may result in reemergence of persistent vaccine serotypes, for example, serotype 19A in Belgium. So continued investment in strong immunization programs is required. So with this, I want to conclude, and I want to thank you, and I'm open for questions.
Thank you so much, Stefanie, for this amazing amount of data, which is sometimes a little bit confusing because there is so much of it. And one of the things that really struck me is, can you speculate on why the serotype 3 increased post-COVID pandemic?
It's serotype 4, you mean, or serotype 3?
I thought it was serotype 3 as well, but
yeah,
4...
serotype 3. Yeah, for serotype 3, yeah, we know that serotype 3, that there's of course some natural variation always also in the serotypes and in, yeah, let's say natural evolution. So this is something that is that we need to take into account. And why serotype 3 increased, I think it was already pre-COVID pandemic that also in other countries serotype 3 did not decrease and increase. So yeah, it's not clear what is the impact of COVID, but on other serotypes like serotype 4 and serotype 12F, yeah, there was the impact really, let's say, where we see after COVID, yeah, different numbers. So for 4, an increase in cases, and for 12F, a decrease in the cases. So I think everybody is still looking into it into more detail to try to explain what is the reason of these, let's say, very special changes in the epidemiology.
Right. Well, thank you for that. So for the audience, please type your questions in the Q&A box, and then I will relay them to Stefanie within the next 5 minutes. The first question, Stefanie, really relates to, it's from an anonymous person. The person is curious why you changed in Belgium from PCV 13 to PCV 10. And I don't know whether you know that answer, but...
Yeah, so I think it's always difficult, and I think it's also interesting to discuss this now, as new vaccines come on the market. These vaccines come on the market based on immunogenicity data. So at that moment, when both PCV13 and PCV10 were available, based on immunogenicity data, it was expected that there would be some cross-protection of the 19F that is included in PCV10, cross-protection to 19A. So there was that evidence regarding immunogenicity data. But I think, yeah, we observed that that cross-protection was not enough to, let's say, to suppress the 19A in our country and led to that re-emergence of that serotype 19A IPD. And this is also something we are confronted with at the moment, also for the higher valency vaccines, is that we have a lot of immunogenicity data, but we have at the moment no idea of the real world impact and what these immunogenicity data will give, let's say, regarding protection for infection, but also, let's say, the impact on carriage, where we need higher antibody levels to impact on pneumococcal carriage. So these are things, yeah, that afterwards it's easy to say, why did you do that? But at that moment, there was some evidence to expect that 19A would be also covered, maybe
based on the surrogate markers that you had,
and also knowing that we had low levels and thinking, okay, we are at that moment on low 19A levels, but you see if the we need to really focus also on these, let's say, vaccine serotypes.
Right. So that leads into the next question from Xavier Lawrence again. PCV15 claims that the immunogenicity against serotype 3 is much better than in PCV 13 and 20. And the person asks, do you expect a better clinical effectiveness with PCV15? And based on your previous answer, I can guess what your answer is going to be.
Yeah, of course. Yeah, there is some evidence regarding higher antibody titers, but we know that also the level that is needed to really protect and also, let's say, prevent carriage of serotype 3 is high. Also, even with PCV15, we see higher antibody titers. It's the question, will it be high enough? And yeah, we will see, but I think personally that we cannot expect that this will have a very big impact on, let's say, our serotype 3 IPD epidemiology, and also depends on to which age group you look. So in children is different than in the adults. Let's say the antibody levels against serotype 3. And for adults, we have no clue what the level really needs to be. So, and as a microbiologist, I can also say serotype 3 is really a particular serotype. It has a very thick capsule, and you can imagine, yeah, it's a very thick capsule. It behaves a bit different than the other serotypes. So it's not surprising that, yeah, this is really a difficult one to handle and also to handle for our immune system. So,
Right.
yeah.
And so a lot of questions do relate to serotype 3. One from Lee Ricketson is, has the serotype 3 severity changed, even though the incidence has not?
I don't know. So the question is really if there is let's say more severe disease by serotype 3 than before. I cannot give an answer. No, I have no idea if if there is a change over time in severity of serotype 3 invasive disease. But as you already know, invasive disease is already severe disease. So I'm not sure if data exists, and if so, yeah, I'm not aware of it. So.
Okay. Considering the epidemiology of countries using PCV10 regarding serotype replacement, should escalation in valency be gradual from PCV10 to PCV13 if the vaccine covers the most prevalent serotypes of invasive pneumococcal disease? A question from Marisol Simus. Basically asking whether the escalation in valency should be gradual. I don't know whether.
I think this question comes a bit from the fact that the higher valence, the higher number of serotypes that are included in a vaccine, maybe the lower serotype-specific immunogenicity is expected, because when you add more serotypes, serotype-specific antibody titers are lower, so we know that. And maybe this question comes from that idea that with PCV13 we will maybe have, let's say, a better protection against, for example, serotype 19A compared to the higher valency vaccines. But also this is, I think, very difficult to estimate. And of course, yeah, it depends also on your local epidemiology. If PCV15 or PCV20 really covers a lot of a high proportion of your serotypes, yeah, then I don't think it's really needed to do that step in between. But sometimes maybe it can be interesting if it's really 19A that is the most important serotype compared to PCV20 PCV13 or PCV15 non-PCV13 serotypes.
Yeah, so there are so many more questions in the Q&A chat. I would like to invite you to take a look at them. I'm sorry that we can't go over all of them, but I would love to thank you for your wonderful presentation, and invite Norman Baylor to share his screen and give his presentation. Norman Baylor has a large background. Many people will know him as a former FDA vaccine review director, and after that he joined the biologics consulting group, where he's today still the chairman, and he's also co-chairman of the Sabin Foundation. Norman, we're looking forward to your talk that will address regulatory considerations for novel serotypes, independent pneumococcal vaccines. Thank you.
Make sure I'm sharing. Again.
Yeah, all good.
Can you see my slides now? Okay.
Yes.
Okay, I need to be able to see them now too. So one moment. Just had them up.
We can see them.
Okay, I cannot, so I need to find them. About that. I'm having trouble finding them now. I know you can see them, but I cannot.
Can you, when you look at, okay, maybe share them indeed again.
Share them again. You should be able to see them now.
Yeah.
Okay, great. Sorry about that.
Okay, well.
Good local time, everyone. Thanks to the organizers for inviting me to participate in this mini symposium. Let's see here. Go to the next slide. Here's my disclosures here. The objective of my talk will be to discuss the potential regulatory challenges of licensing a non-capsular pneumococcal vaccine, considering that we already have very effective and safe PCV vaccines. Just to provide a little background here, despite the successes of the PCVs, the current PCVs that we have, as you've heard from other speakers, there are limitations to these vaccines. And we know with the emergence of non-vaccine serotypes contributing to carriage and disease in high PCV coverage settings, there's also the complexity of designing these vaccines from a chemistry, manufacturing, and control perspective when there are multiple capsular polysaccharides. Additionally, there are high costs to the PCVs, and that limits the access of these vaccines to, or potentially limiting the access of these vaccines to developing countries where the burden of disease remains the highest. And so protein vaccines offer the potential of extending the serotype coverage and improving protection against such diseases as otitis media and pneumococcal pneumonia. Currently, there are at least, these are the licensed pneumococcal vaccines. There are 4 pneumococcal vaccines available in the United States, and these vaccines include the PCV15, the PCV20, which you've heard about today, and then recently the PCV21, which was licensed in the third quarter of last year. And then there's, of course, the pneumococcal polysaccharide vaccine for adults, and this vaccine was licensed many years ago. So just for those of you who really may not have a lot of background on the regulatory pathway, the Food and Drug Administration in the U.S., the licensure pathway of vaccines are generally, there are three broad licensure pathways, and each of these have different approaches for demonstrating effectiveness with the same requirements for demonstrating safety and manufacturing consistency across all three of these approaches. There's the traditional approval, where looking at endpoint that directly demonstrates a clinical benefit. And then there's accelerated approval, where there's a surrogate endpoint that's reasonably likely to predict a clinical benefit. And then thirdly, there's the animal approval, where when the other two approval pathways are not reasonable or not possible to use, one may consider the animal rule approval, but I always tell folks that the animal rule is much more difficult than the other two pathways. So in reality, for these two, for pneumococcal vaccines, it would be traditional approval or accelerated approval. And so the licensing strategy for PCVs was the demonstration of safety and a statistical immunological non-inferiority of the new vaccine with the currently licensed products. And this started with the PCV7 with the licensure of PCV7, and that was the comparator in many of the new vaccines. PCV7, of course, had an efficacy study to support, had efficacy studies to support licensure. And then there's the effectiveness of conjugate-based vaccines against IPD in different populations, assessed by comparing the immunogenicity of each serotype versus a previously licensed vaccine. So those are the two strategies that have been used for licensing the PCVs in the past. Since there's no correlate or established immune correlate of protection for prevention of IPD in adults, the demonstration of non-statistical non-inferiority of OPA titers for traditional approval of new higher valency pneumococcal vaccines for the prevention of IPD caused by the vaccines and the serotypes. However, immunogenicity alone is insufficient to support a non-invasive disease indication, such as pneumonia for, in this example, the 7 new non-PCV13 serotypes included in PCV20, and then now the 8 new the serotypes in the PCV21, because the antibody levels have not been found to be indicative of prevention of non-invasive pneumococcal disease. And then, as I mentioned to you about accelerated approval, the way some of these vaccines have been licensed based on a surrogate endpoint, the OPA titer is an established immunological surrogate endpoint that's reasonably likely to predict prevention of pneumococcal pneumonia caused by new vaccine serotypes. And so pneumococcal pneumonia it's a serious condition, and PCV20, PCV21 is intended to provide a meaningful therapeutic benefit to patients over existing treatments. And so that's the criteria, one of the criteria for accelerated approval by the US FDA. So, however, as a condition of accelerated approval, the US FDA has accepted a proposal for conduct of a post-approval real-world observational test negative case control effectiveness study as a confirmatory study to verify and describe the clinical benefit of prevention of pneumonia in adults caused by 7 new serotypes. And so this is a post-licensure commitment, and this is also the same commitment that was required for the licensure of the PCV21 as well. So now turning to pneumococcal protein vaccines, these protein-based vaccines have emerged as a promising strategy to address the limitations of the polysaccharide-based vaccines. Unlike the polysaccharide capsules, which exhibit significant antigenic variation among different serotypes, certain of the surface proteins of strep pneumoniae are more conserved and are shared across serotypes. So by targeting these conserved proteins, protein-based vaccines have the capacity to offer broader protection against a wider range of pneumococcal strains, including those that may not be covered with the PCVs. The development of pneumococcal protein vaccines have been fueled by the advancements in understanding the biology and the pathogenesis of S. pneumoniae. And so the identification of key virulence factors and surface proteins involved in host pathogen interactions has facilitated the selection of potential vaccine candidates. On the other hand, and most of you know, whole cell vaccines use the entire cell of the bacterium. They present all protein antigens without the need for individual protein purification to stimulate the immune response. And they can be developed from killed or live attenuated whole cells or by using genetically modified strains of the bacterium to ensure safety while retaining immunogenicity. These vaccines their approach aim to induce both humoral as well as cellular protective immune response against multiple antigens of the bacterial cell in at least in animal models. And these vaccines have also been included in include a variety of strains or serotypes of the bacteria, allowing them to cover a broader spectrum of pneumococcal infection. So this is the attractiveness to both whole cell and protein-based vaccines in comparison to the PCVs. Now, protein vaccines as a potential vaccine candidates, there are numerous protein candidates have been investigated for the potential of vaccine antigens, such as those I have listed here. These proteins have been shown have shown promising immunogenicity and protective efficacy in animal studies, and some of these candidates also induce immune responses that inhibit bacterial adherence, colonization, and invasion. Several of these proteins have been evaluated in early clinical studies, and they provided insights into their safety and immunogenicity in humans. The most reasonable approach probably is combining these multiple protein antigens in vaccine formulation as opposed to trying to move these vaccines forward given the challenges as a standalone vaccine. So either using these multiple protein antigens either as a multi-component vaccine or using carrier proteins may potentially broaden the serotype coverage and enhance the protective immune response. And the high degree of antigenic variability among pneumococcal strains necessitates the identification of conserved epitopes and the design of vaccines that provide a broader protection against diverse serotypes. Now, proteins as standalone vaccines, while they are less complex and potentially cheaper to manufacture than PCVs, a standalone multivalent protein approach has an undefined regulatory pathway. It will probably require human efficacy data for licensure, and this is a challenge in itself, given the size of studies needed that may be needed to determine the efficacy of these protein-based vaccines. And these vaccines will probably need to demonstrate a reduction in nasopharyngeal carriage, as well as what their impact will be on herd protection. And these vaccines, protein-based vaccines, may not be as efficacious as our current vaccines, though it's maybe the advantage may be the potential for broader protection against all serotypes. This might compensate for being less efficacious than PCV. And then, although the correlates of protection are often unknown, they are not required for licensure. So even though we don't have correlates of protection for those. Identifying that correlate is not in itself a requirement for licensure. So there are really more questions than answers, because if we look at the protein-based pneumococcal vaccines, I mean, from a regulatory perspective, it begs the question: what indication are you looking at for these protein vaccines? Is it invasive pneumococcal disease? Is it pneumonia? Are we seeking pneumococcal whole cell or a protein vaccine? And then what is the surrogate marker? Is there a surrogate marker that is reasonably likely to demonstrate clinical benefit from, again, looking at standalone with these protein vaccines? And then a very provocative question is over time has been, can carriage serve as a surrogate marker to be confirmed in a post-marketing clinical study? And this has been asked of regulators, although at this time, carriage is an indirect effect of vaccination, and the indirect effects of vaccination are not currently established as part of the licensing process. So, what is the contribution of that vaccine against carriage, for example? And then, is there a role for human pneumococcal challenge models using prevention of carriage as a proof of concept? So these are questions and considerations that are wrestled with by the regulators, as well as the manufacturers of these novel investigational vaccines, if you will. And then another question would be, can cellular immunity, in addition to IgE in serum and specific antibodies, contribute to an understanding of the mechanism of protection of vaccines and support licensure? Again, these are questions that the developers and regulators are trying to obtain answers. So, in conclusion, the current regulatory strategy for PCVs may not support licensure of a protein or other mechanism, novel mechanism vaccines, for the reasons I've articulated earlier in the talk. So, evaluating the efficacy of new PCV products against disease endpoints has been extremely difficult, partly due to the potential ethical consideration as well, given the sample size requirements, and also given the fact that the current vaccines have done so well and are highly efficacious and safe. And so what may help is identification of correlates of protection and surrogate endpoints. These might facilitate regulatory strategy for licensure of next generation pneumococcal vaccines. But I'll reiterate that probably the role for the protein-based pneumococcal vaccines is in combination with what is currently there, or in combination, looking at some of the proteins in combination. But again, some of these studies may be required to be very large, and are those type of studies feasible for developers? And I think that is the end. Yes, so I'd be glad to take some questions.
Thank you very much, Norman. As usual, FDA might leave you with more questions than answers when listening to this very complex matter. You clearly outlined that there is a need for better vaccines. That was also already indicated in the talk by Dr. Desmet. And I'm just wondering, given where we are and what we know right now, what would you recommend to all those smaller companies that are trying to develop a universal pneumococcal vaccine? I know you tried to summarize that in your last slide, but just summarize it for us once more.
Okay, sure. I mean, it's very difficult to move these. I mean, there are many of these protein-based vaccines in clinical studies, in trials, but the difficulty is trying to determine the efficacy and the effectiveness of these vaccines against what we already have, the current vaccines. And I think that's a challenge. I mean, if efficacy studies are going to be required for these, or even if you're looking at the potential of non-inferiority, these are going to be large studies, and it begs the question: can these vaccines do they have the breadth to protect as well as the current vaccines? Pneumococcal conjugate vaccines have done so well, although, again, as I said earlier in the talk, the serotype replacement and the cost, and I think even back to the first talk, you met with Kathleen. You asked the question, well, how far can we go? I mean, there are hundreds of serotypes for pneumococcus, and it's difficult. It's very difficult, that conjugation process. I mean, we're up to 21 now, and how far are we going? How far from a chemistry manufacturing control process can we go adding serotypes? However, if you can find a protein or a set of proteins that are conserved across the serotypes, that might be the way to go. So either a combination, adding conserved proteins to the current vaccines may be the best approach, given what the challenges of trying to license these vaccines as standalone.
Yeah, I recall that GlaxoSmithKline did add at some point in time 2 proteins that were, highly promising to their vaccine, and did some very large efficacy studies. But apparently that program didn't move forward, probably because exactly of the reasons that you described, that it's so hard to demonstrate in the field what
and what indication are you looking at?
Right.
So.
Yeah. I think I do not see at this moment in time specific questions for you in the chat. So what I would like to do is I would like to thank you for your time for this overview. Again, gives people a lot of stuff to think about, and if more questions come in, I'll reach out to you later again. Thank you very much, Norman.
And I will stay on the seminar just in case things come up. So.
Thank you. And then I would like to invite Cynthia, Dr. Whitney, who has a long history of working with the CDC and also being involved in many, I think, epidemiological studies and data, but who is nowadays the president of the ISPPD, the International Society that really deals with pneumococcal disease and the prevention thereof. And she's going to give us an overview of what the remaining questions are around pneumococcal vaccines and pneumonia prevention, and will give us a perspective from the International Society of Pneumonia and Pneumococcal Diseases. And I think a lot of issues were raised, Cynthia, and there's nobody better than you to help us address those. Thank you for participating.
Thank you for the invitation, Manon. Let me put my slides on here.
Yep,
can everybody see the slideshow mode?
We can see the slideshow. Thank you.
Okay, so thank you for that nice introduction. As you heard, I was at CDC for many years working on pneumococcal vaccine policy and epidemiology. And for the last 5 years, I've been at Emory University and have been running a program called Child Health and Mortality Prevention Surveillance, or CHAMPS. And it's given me sort of an interesting perspective on pneumonia, as has been becoming the president of ISPPD. So I'll talk about some of what I think are the remaining questions. There's plenty of areas that need ongoing research, and I'm excited to be part of this discussion today about how pneumococcal vaccines can be are an important part of that. So let me start with a case example that we have from the CHAMPS project. Now, CHAMPS is an unusual project. We work in 9 countries, Sub-Saharan Africa and South Asia, and we set up in communities and work intensely to identify stillbirths and child deaths in real time. And if we can contact the families before the burial takes place, we get consent to actually take postmortem specimens. So we do PCR, we do culture, we look at the pathology of the tissues. So we actually get a very good idea of what happened to those kids. So it's a unique model that I think gives us some interesting insights about what happens in high mortality settings. So with this case, it was a death we identified, an 11-month-old boy. He'd been coughing for 3 days, stopped eating a couple days before, and then the day of his hospital admission, he developed fever. And he had been receiving some traditional medicines at home with no improvement. By the time he arrived at hospital, he was already in respiratory distress. He was drowsy and in poor condition, and the medical professionals started treatment, but the boy died within 24 hours of arriving at the hospital. So he was enrolled into CHAMPS, and our team took postmortem measurements that showed that the child was severely malnourished. Usually a Z-score of less than -2 is malnourished, and you can see these are much less than that. He was 11-month-old and only weighed 5.5 kilograms. The testing was collected only 2 hours after the baby died, so there's not many concerns about postmortem contamination or overgrowth. On our testing, we didn't find any gross abnormalities that were seen. HIV, TB, and malaria tests were negative. The blood culture grew Klebsiella pneumoniae, and we did ATTACK is our PCR platform, and the blood and CSF were negative, but you see in the lung tissue that there were all sorts of viruses and bacteria listed here, including pneumococcus and Klebsiella. The pathology of the liver was unremarkable, but the lung showed strong evidence of bronchopneumonia. And when we did our special immunohistochemical staining, there's evidence of Klebsiella and pneumococcal infections. And here's the tissue specimens that show this. So here's on the left is lung with the purple is Klebsiella pneumoniae that are lighting up with immunohistochemistry. And when you look at the liver tissue on the right, you can see the pneumococcus lighting up with IHC. So this baby clearly had malnutrition as an underlying condition that unfortunately set him up for this kind of severe pneumonia where Klebsiella and pneumococcus worked together. So I think this case illustrates a lot of things. One, that there's a lot of kids out there that have conditions that set them up for severe pneumonia. And also pneumococcus certainly can cause bad disease on its own, but often that it works with other pathogens. So what else can we do to reduce pneumonia illnesses and death? I think we need to consider addressing some of these factors like malnutrition that not only reduce vaccine responses, but also increase risk for pneumonia, or if a child or an adult gets pneumonia, they have a poor outcome like death. A lot can be done to improve diagnosis and diagnosis and treatment, both access to diagnostic tests and good quality treatment. But we also need to consider how co-pathogens might be working with pneumococcus to lead to poor outcomes. For vaccines, there's a lot of things we can work on. The sustainability, especially in low and middle income countries, improving the supply. And I think we can do more to maximize the effectiveness against pneumococcal disease. And we've talked a lot about maybe what serotypes should be in these things and how many can we include. For pneumonia holistically or pneumococcal disease, I think products that address other pathogens like Klebsiella could be adjunctive programs that actually end up reducing pneumococcal deaths because of the way that these pathogens work together. And I think we can evaluate and adjust vaccine schedules and think about not only children, but adults. So let me dive a little bit more into some of these issues. Here's a slide from CHAMPS that shows what kind of conditions are common in children who die of pneumonia. we find pneumonia a lot in our infant and child deaths. It's in about 40% of those we evaluate. The median age at death is about 9 months with an interquartile range of 4 to 19 months. 2/3 of the pneumonia deaths we enroll are in the community, but there's a lot actually that appear to be hospital associated, where kids come into the hospital with one thing, and if they stay long enough, they get a bad pneumonia. And what we see is not healthy kids who are outside playing and come down with pneumonia and die. These are kids that have malnutrition, HIV, sepsis, congenital defects. The figure on the right is from one of our manuscripts that shows these. You can see the high proportion of deaths that have malnutrition and surprisingly HIV. We also see conditions like malaria occurring with pneumococcal disease. And I think, as challenging as this may sound, that all these things are occurring together, it does highlight that there are opportunities to intervene. If you can prevent HIV, you're going to absolutely reduce pneumococcal disease risk. We can do a lot more to improve treatment. In these kids who died, less than half were treated with oxygen, either because of late arrival to a medical facility, misdiagnosis, no measurement of the oxygen level, or simply a inadequate oxygen supply. Now, one of the things that I find fascinating about our CHAMPS data is that because we have lung tissue, you really can get a good sense of the etiologies of pneumonia deaths. And, we do a lot of testing, but the people that review the cases only look at, okay, which pathogens really seem to be causing the pneumonia. But when they do that, we still come up that in general, there's 2 or more. The median number of attributed etiologies is 2. But we do find a lot of pathogens in the lungs of, over 1,200 positive PCR tests on lung tissue. Only about 40% are considered causal. The others appear to be, bystanders that are detected. So postmortem lung sampling is a high yield for diagnosis. In over 80% of our deaths, we can identify causative pathogens. And in most of those, multiple pathogens are implicated. And this table on the right shows you the most common pathogens. Klebsiella is the #1 detected. And you might be thinking to yourself, what? That's not what pneumonia papers usually say. But remember that these are all children who have died. So these are people that have failed treatment. So Klebsiella is often resistant to amoxicillin and can be resistant to 3rd-generation cephalosporins. So these are kids that are failing therapy. And often they're occurring in combination with pneumococcus, like the case I showed you. So pneumococcus is clearly remains a common cause of pneumonia deaths in spite of vaccination programs existing in all the countries where we work. When we look at the pneumococcal serotypes in those specimens where we can, at least 80% are non-vaccine serotypes, as you might expect. We also have a lot of CMV causing pneumonia, which reflects, again, the kind of children who can't survive a pneumonia episode, a lot of malnourished kids, a lot of kids with HIV. So when you look at these data, I think there's, some ideas here for how vaccines can improve this situation and help save lives. Clearly, a vaccine against Klebsiella pneumoniae would help. And for those vaccine manufacturers in the crowd, I would encourage that pursuit of that type of vaccine, not only for this indication, but for all of the healthcare-associated infections that are out there. And clearly, pneumococcal vaccines that cover more serotypes might also help. And if we can expand programs on use of RSV vaccines and other kinds of treatments, that would help as well. So we've been talking about serotypes. How do we figure out which serotypes to include in our new vaccine formulations? Well, I think the Serenade project that was discussed earlier does give us some ideas. Clearly, this project showed that serotype distributions have changed as a result of effective vaccination programs. It adds to the data showing that 19A was one that could emerge when other vaccine serotypes were taken out of circulation. Serotype 3, as we've discussed, remains a challenge. And if we add more serotypes to vaccine formulations, this does increase the proportion of IPD cases that are potentially preventable. But, which ones to choose? And I'm not going to give you any answers today because the individual remaining types cause a small proportion of disease. It's also a challenge to think about, whether this is a static picture. Will serotypes that are rare now become more problematic? And I don't think we really understand when and why this can happen. But in theory, it could. So, the discussion about common antigen products is attractive, but, as Norman said, there's some challenges with work to develop those kind of novel approaches. But I think they're important to pursue. Vaccine schedules is a hot topic. Pneumococcal conjugate vaccines have a complex design. Each of the serotypes is really its own vaccine. And that makes these relatively expensive compared to some of the simpler and older products. More doses in a schedule equals more costs, equals more concern over sustainability. As we heard, most countries do use a 3-dose schedule, either 2 doses early on with a booster at around a year of age, or 3 early on without a booster, which is a common schedule in low-income countries. The US and a few others are sticking to their 3+1 because that's what they started with. But usually most countries in the world are on a 3-dose schedule. We know these schedules are highly effective at reducing transmission and inducing herd effects. So can we use fewer vaccine doses, or will that mean more disease? People are talking about 2 doses, maybe a 2-dose schedule that focuses on the booster as generating herd effects and sustaining disease control. When you vaccinate toddlers with 1 dose, how much herd effect can that generate? Can you split doses in half? I mean, these products are so effective. Do we need as much antigen in each dose? I think these are all interesting questions that people are researching, but pneumococcus is everywhere and highly transmissible. So we have to make sure that we're not dropping so many doses or dropping so much antigen that we're leading to more disease and ineffective programs. So important things to research, but definitely open question. Another thing we need to keep working on is to reaching those kids that are yet remain unvaccinated. This is a slide from IVAX View Hub that shows the number of unvaccinated kids. You can see there are some big countries like the US and Brazil, with 1 million or more kids that are unvaccinated. I mean, the coverage in the US is pretty high, but you still, even if you're missing 10% of kids, it results in a lot of missed doses. And you can see there are some countries that are still not using product. And in parts of Africa, there's just hard to reach populations. So the product characteristics, the cost, the supply, the cold chain requirements do affect how many kids can be reached. So I encourage those working on vaccine development and evaluation to consider these factors as they develop their products. So what about adults? Adults in many places are living longer, and we know that as you age, your risk of pneumonia increases, your risk of IPD increases because of immune senescence. And as you age, you not only have a higher risk of disease, but a higher risk of not surviving your infection. We also know that adults with certain chronic or immunosuppressing illnesses are highly susceptible to pneumococcal disease. And we know that adults have a much more diverse serotype profile than children. So some of the newer products are a much better fit and much more effective than the old polysaccharide vaccine or, the early seven-valent generated products. So you heard about the new US program recommending it down to adults to age 50. And I think probably more countries out there are considering vaccination of adults for pneumococcal disease now. But adults live a long time. So when do you vaccinate? Does efficacy drop with age? If you vaccinated age 50, when are you going to revaccinate? And what is the cost effectiveness of all of this, especially if you've got a pediatric program that is reducing the transmission of some of the most invasive serotypes for adults. So a lot of questions remaining about adult immunization programs. So let me shift gears a little bit and talk about ISPPD, or the International Society of Pneumonia and Pneumococcal Diseases. It was launched in 2012 because we needed a framework for the International Symposium of pneumococci and pneumococcal diseases that was becoming increasingly popular. The mission of the society is to improve the knowledge and understanding of pneumonia biology, the pathogenesis and epidemiology, the host response to important pneumonia pathogens that are carried in the respiratory tract, and how to better prevent and treat pneumonia. So it was really to bring together interested people so we can speed the research in all of these areas. So our objectives include facilitating scientific exchange through the biennial every 2-year International Symposium, and to support other symposia and educational activities like the one today that support our mission of advancing research in this field. We want to promote scientific collaboration and research, and to promote public awareness of pneumococcal disease treatment and prevention. So I'm the president. David Goldblatt at the University College of London is the secretary, and Debbie Bogart, the University of Edinburgh, is our treasurer. And if you go to our website, we have a stellar set of ISPPD board members that help drive the agenda for the society. You can be a member of ISPPD. It's free to join. If you're a member, you can apply for research awards that support projects for junior researchers with starter grants. We have travel awards to support conference attendance for those from low and middle income countries, and there's a reduced cost for conference registration. So go to the website and join us. Our next meeting will be in Copenhagen in May of 2026. I hope people can come. The meetings usually have somewhere between 1,000 and 1,300 attendees. The organizers work hard on an exciting program that covers original research, but also has expert lectures like the great ones you heard today, as well as workshops to train on specific issues. And we have fantastic social events. So I hope you can join us in Copenhagen in May of 2026. So in conclusion, it's clear that pneumococcal vaccines are showing remarkable benefits, but work remains to reach everybody. Too many children and high-risk adults are still suffering from pneumonia and other pneumococcal diseases. A lot of promising new products out there. Some are still in the evaluation stage, some are in the rollout stage. So we're excited to see how these work, but how will pneumococcus evolve in response to these new vaccines? We need to better understand also the interactions with other pathogens. But I'm pleased to say that there is a robust research community working on these and other questions that's coordinated through ISPPD. And I am very grateful for that. So I will stop there and see if anybody has any questions.
Thank you, Cynthia, for a beautiful overview. Again, I was actually, well, first of all, I want to say I did attend your last ISPPD meeting, and I totally agree. Fantastic program and also great social activities. It's really a wonderful meeting. So I'd also highly encourage people to attend that meeting. I was encouraged to see that you mentioned viruses, RSV, as one of the things to think about, because as a former and actually still very interested in influenza viruses as a vaccinologist, what people that are virologists always say is it's the viruses that cause the tissue damage that allows these bacteria that are always in and around us to really cause invasive disease. And I would love to hear your perspective of it. I can imagine that mentioning RSV probably goes into that direction, but influenza, COVID, all those other viruses that are so predominant, what are you guys doing about those?
Well, it was funny when I worked at CDC, I would always argue in jest with my flu colleagues and my RSV colleagues about, why don't they come work on a real pathogen like pneumococcus? But it was very much a friendly argument. But over time, I just see over and over how these things work together. You get a virus, you're coughing, you're sneezing, helps you spread whatever's in your upper airway to somebody else. Maybe even it goes down. Maybe it changes the concentration. You get your pneumococcus is happily sitting there. Flu comes along and messes with your microbiome. You start coughing, you get an infection. I think these things are working together more than we know. And it would be wonderful to understand that. I mean, there's been some fascinating publications. I remember Shabir Mahdi and Keith Klugman, when they were doing some of the PCV9 work in South Africa, how they showed that, pneumococcal vaccine prevents influenza hospitalizations. I mean, these things work together, and when they're together, they're worse.
Yeah. Yeah, I think it is fascinating. So definitely more research is needed to really address these situations. I do not see at this moment in time any specific questions for you in the chat. I think that you gave us a lot of material to think about. Also the Klebsiella, really interesting, and it's clear that the antibiotics play an important role there, right? If they don't work, then clearly you can't prevent that bacteria. But I was wondering one more question around your CHAMP study. Those kids that died, what percentage of them was actually vaccinated? Do you know that?
That's a good question. It's hard to know exactly because a lot of times by the time a child dies, the vaccine card is long gone. In the countries where we work, the coverage is typical for African settings, probably 70-80% have 3 or more doses. But again, we're getting the kids who fall through the cracks.
The question would there be a much higher percentage of non-vaccinated kids there, right? That would be, I think, worthwhile identifying.
Yeah, no, we definitely have. I'm sure that plays a role. We're not able to get that data exactly, but it's interesting when we look at the serotypes that there are 80% non-vaccine serotypes. So the programs in those countries are changing transmission. And in spite of that, kids are still dying, from pneumococcal pneumonia, often with other pathogens too. So it's not all under vaccination. It's also that the products can't cover everything, and that these kids are so ill from malnutrition and other things that sometimes even if they are vaccinated, did that vaccine have a chance in an HIV kid or a kid with malnutrition to really do its best work?
Right.
So Yeah.
All right. Well, I would like to thank you very much for your overview. I really enjoyed it, and I would like to close out this this seminar with giving a couple of slides on our organization, the International Society of Vaccines. So thank you first of all to all our speakers, and I'm going to try to share my screen now to come back to some of the slides that I would like to close out with today. Let me take a look how I can. Yes, so the International Society of Vaccines does more than organizing this mini-symposium. We also have other seminars that we give, and the next seminar that we'll be giving is in principle a seminar that is set up specifically for the next generation scientists and next generation vaccinologists, I should say. And what this seminar tries to do is we try to have a more experienced vaccinologist in combination with a younger scientist give an overview of their career and things that they're doing at this moment in time. And the next one is going to happen later in February. We also have a series where we speak about topics of interest, monthly webinars, and the next monthly webinar will take place on the 25th of February with Hulan Shen, who will actually speak about, sorry about that, who will actually speak about emerging avian infections and vaccines. So I invite everyone to participate in that if you're interested. The ISV organizes an annual congress, and our next annual congress, 225, will take place in Stellenbosch, October 28th to October 30th. I really hope that we're going to have some of the participants that were here today speaking, some of the speakers of today attending that conference as well, so that we can dive a little deeper into pneumococcal disease and pneumonia. The ISV organization is also a membership organization with a lot of benefits previously pointed out, but we have reduced registration fees for our Congress. We have access to career mentorship programs, and I have unfortunately this presentation on automatic move forward, so it goes a little faster than I want. You can participate in our committees. We have regular newsletters. We offer opportunities to network, and we publish monthly papers of the year, monthly papers and paper of the years. You are able to get free access to the online journal of vaccine when requested, and discount on human vaccines and immunotherapeutics as a member. The ISV Industry Committee was established to ensure the sustainability of our society, and I really want to thank our collaborators and our sponsors for their sponsorship, because that allows us to do these seminars and to really get the society together and get people together to, share experiences. And the schedule that we've put together for our quarterly mini-symposias are listed below. We will have the next one on RSV vaccines on April 24th. On June 26th, we are scheduling HIV vaccines. September 25th, influenza vaccines. And we are open to work on many different topics. And please email me at manoncox@nextwavebio.com to share your ideas and share topics of interest to you. And again, then I would like to conclude with thanking our speakers and of course also our audience for attending today, for your very active questions. And again, we look forward to seeing more participation and engagement at our next meetings. And again, you can follow the International Society for Vaccines on Facebook, LinkedIn, or YouTube, and there's a nice QR code for you to get connected. So with that, I would like to close this seminar and thank everybody for being here today, and especially our speakers. Thank you. Have a wonderful day.